Welcome to part 2 of this 10-part online training program on adaptive designs and clinical trial simulation. In part 1, we went over general objectives of adaptive designs in clinical trials, and then I gave examples of adaptive designs that are used in proof of concept trials, also known as phase 2 trials. And we focused on a very appealing class of designs that support response adaptive randomization. And once again, even though I refer to clinical trial examples in the context of pharmaceutical drug development, the general framework presented in this online training course is applicable to a very broad class of studies that are conducted to evaluate the efficacy and effectiveness of vaccines, of general interventions in a randomized setting. The only fundamental assumption that we're making here is that we're dealing with randomized trials. I would like to point out that this is an introductory module. In this module, I'm only going to emphasize key ideas behind the individual adaptive designs. We will be aiming at conceptual understanding, and all of the technical details, statistical details, information on the relevant software tools will be presented in parts 3, 4, all the way up to part 10. In this module, we will discuss adaptive designs that arise in the context of confirmatory trials or phase 3 clinical trials. I will begin by pointing out that while there are multiple examples of adaptive approaches that could be considered in dose-finding trials, there is clearly a lot more diversity in terms of different types of adaptation, different types of statistical rules, decision rules that could be applied when it comes to a confirmatory setting in clinical trials. And in this training program, I'm going to highlight 3 most common types of adaptive rules, 3 essential distinct types of adaptive designs. I think it's going to be helpful to begin this discussion by introducing motivating examples, case studies that are based on real-life clinical trials that have taken advantage of various types of adaptive design features. These case studies are quickly presented on the slide, as well as the next slide, and they will help us prepare for a detailed overview of each class of adaptive designs. We will first take a closer look at adaptive designs that support sample size re-estimation or event count re-estimation if this happens to be an event-driven design. The main idea behind this particular type of adaptive designs is that the interim data are examined, and the interim data could be potentially used to improve the overall probability of success in the trial by increasing the target number of patients or events in the trial. We will use as a motivating example for this class of adaptive designs the VALOR trial in patients with acute myeloid leukemia that was published in 2015, and that will illustrate an adaptive approach to using interim analysis data to increase the target number of events in this oncology trial. The next class of trials, which is also quite popular but perhaps slightly less popular, is a class of adaptive trial designs that support adaptive, data-driven treatment selection at the time of an interim analysis. Here, the interim data would be examined to identify the most promising, the most effective treatments, or one or multiple treatments for the final assessment in the trial. And this class of adaptive designs will be illustrated using the ENHANCE trial in patients with chronic obstructive pulmonary disease that was published in 2010. And this may be, most likely, it is the very first phase 3 confirmatory trial that utilized this adaptive approach with an option to select the most promising treatments at an interim analysis. The last class of adaptive designs that will be considered in this online training program in the context of confirmatory clinical trials would be adaptive designs with data-driven population selection. In this case, the goal is to examine the interim data and to understand whether the most promising patient population or populations could be selected for the final assessment, or if the trial could perhaps continue as originally planned to the final analysis. We will use the Saturn trial. That was an example, one of the first examples of a multi-population trial. It was conducted in patients with advanced non-small cell lung cancer. The results were published in 2010. And even though this was not technically an adaptive design, I will explain to you how this particular trial could have actually benefited significantly by employing certain data-driven decision rules that would help identify the best patient population for the final analysis. In this section, we will provide a review of regulatory requirements for adaptive designs used in phase 3 or confirmatory trials. In this section, once again, I will talk about key regulatory requirements for adaptive designs in a confirmatory setting. We will go over 4 key requirements for adaptive phase 3 trials. And although it is common to think about these regulatory documents as regulatory guidelines, these are of course based on solid scientific principles, which means that they represent important scientific guidelines, and therefore it is important to follow these scientific guidelines when we design, conduct, and interpret the results of adaptive trials. And we can refer, for example, to the guidelines published by the United States Food and Drug Administration. I referred to this guidance document in part 1, and at that time I emphasized that this regulatory guidance document fully endorses adaptive trial designs that enable various data-driven decision rules, for example, futility stopping, for example, sample size, re-estimation, treatment, and population selection. We just talked about those most popular types of adaptive decision rules a minute ago. And this guidance document also formulates 4 key requirements for adaptive confirmatory trials. These requirements are related to the 4 topics shown on the slide. I'm going to present them very quickly right now, and we will go over them in detail later in this section over the next several slides. And as you can see here, the 4 topics of most regulatory and scientific significance in the context of adaptive designs deal with the requirements to control the likelihood of erroneous, incorrect conclusions, and that likelihood, that probability, should be adequately controlled. Secondly, the trial sponsor is expected to consider relevant statistical methods that provide reliable estimates of the treatment effects, treatment differences. The third requirement deals with the standard requirement that all of the elements of an adaptive design must be completely pre-specified, must be pre-planned. And finally, requirement number 4 refers to the fact that a sponsor of an adaptive trial is expected to take all of the necessary steps to ensure trial integrity. So let us begin with the first requirement, or requirement A. First of all, it is mandatory, as I have said before, to ensure type 1 error rate control, or it's also known as alpha control or the false positive error rate control, in any confirmatory trial that would be used for regulatory approval purposes. And an important feature of data-dependent changes that are based on unblinded interim analysis data, and that's one of the key features, of course, of any adaptive trial, is that those data-dependent changes are very likely to contribute to type 1 error rate inflation, And it's not then surprising that regulatory agencies, and as I said a minute ago, not just regulatory guidelines, it's about general scientific principles. Those general scientific principles require that appropriate statistical methods should be applied to guarantee to retain type 1 error rate control in the context of adaptive trials, where once again certain decisions, certain elements of the design could be updated on the fly, if you will, based on the data available to the sponsor at an interim assessment. And I think it would be helpful to give a quick example. And it is well known, for example, that the alpha is likely to be inflated in trials that employ several looks at the data, interim looks or interim analysis, where the trial sponsor could claim a statistically significant benefit for the experimental treatment. Those are commonly known as group sequential trial designs. And why does this happen? In very simple terms, it happens because there are multiple shots on goal, which means that there are multiple opportunities to potentially claim a statistically significant benefit in the trial simply by chance, when in reality, when in truth, the treatment or intervention of interest is not effective, and that leads to the inflation of the alpha level or of the type 1 error rate. And the same logic also applies to adaptive selection of patient populations, adaptive re-estimation of the sample size. It is a bit more subtle, the sample size re-estimation, but it's also the case that the alpha will get inflated if unblinded interim analysis data are used to support a decision to expand the trial and enroll more patients, or perhaps increase the target number of events if it happens to be a survival trial. The good news is that statistical methodology that guarantees overall alpha control in an adaptive trial, regardless of the type of design modifications, exists. And this methodology relies on the so-called combination function principle that has found numerous applications in adaptive confirmatory trials. We will talk about these principles a little bit later in this module. And for now, we're going to continue to the next requirement, or requirement B. And this one deals with the fact that an estimate of the treatment difference, for example, the mean treatment difference if the primary endpoint is continuous, or the hazard ratio if the primary endpoint is a survival type endpoint, so that estimate is subject to bias when data-driven decisions are implemented in a trial. And this bias considerations also apply to the confidence interval for the treatment effect. What happens here is that the naive point estimate and the confidence interval become unreliable in the context of adaptive trials that once again rely on the data collected in the trial to update the trial's design, and therefore they should not be used at the final analysis if design was modified at an unblinded interim analysis in the trial. Again, statistical methods exists. They have developed to compute the so-called bias-adjusted estimates of the treatment difference, as well as bias-adjusted confidence intervals for the treatment difference. And that means that a statistical adjustment or correction will be applied to have this bias induced by the analysis of by the use of data-driven decision rules. That bias will be removed, and for this reason, bias-adjusted estimates of the treatment effect and confidence intervals are recommended for use in adaptive trials. Continuing to requirements C and D, those are somewhat related to each other, and we will discuss them at the same time. The FDA guidance states, for example, that it is imperative to maintain trial integrity and reduce operational bias in adaptive trials. The term operational bias is used a lot by regulatory reviewers, and it refers to the fact that when we have the exact knowledge of interim analysis results, and that information is disseminated to the trial investigators, it can influence their behavior. It can even influence patients, and even the sponsor's decision-making process could be influenced in potentially unreliable ways, which is what makes it difficult. We know that this will affect the credibility and ultimately interpretability of the trial's results. And that means that dissemination of unblinded interim analysis results must be avoided, and only members of the data monitoring committee that will review unblinded data, and they will make recommendations as to what specific data-driven decisions will be made for the remainder of the trial. Only those individuals should have access to unblinded interim analysis data. This is the most important aspect of maintaining trial integrity. And in general, to help trial sponsors maintain trial integrity and reduce operational bias, the regulatory guidelines, the FDA as well as the EMA guidelines on adaptive designs provide a list of important points that need to be considered by sponsors of adaptive trials. The most important requirement is the need for prospectively defined standard operating procedures for implementing adaptive trials. I will give you a couple of examples. For example, the sponsor needs to demonstrate that access to interim analysis results will be limited once again to the Independent Data Monitoring Committee, as well as the Independent Data Analysis Center, which is the group of individuals who will be analyzing the unblinded data and then presenting them to the Data Monitoring Committee. It will be important to set up the so-called Data Monitoring Committee charter, create a data access plan, a communication plan between the Data Monitoring Committee and the sponsor. All of that needs to be detailed in the Data Monitoring Committee charter. It will be important also, it will be critical to establish firewalls to prevent unauthorized access to the unblinded interim analysis results and recommendations made by the independent committee, the Data Monitoring Committee. The sponsor would be expected to take additional steps to ensure that trial investigators cannot infer, they cannot back calculate the treatment effects or other interim analysis results based on the decisions that the sponsor will share with the investigators. And here I'm going to jump ahead a little bit. We will definitely come back to this very important topic of trial integrity in the modules on adaptive designs and confirmatory trials, for example, in part 5, when we will discuss statistical rules for sample size re-estimation. We will introduce a rule that makes it impossible to back calculate, to reverse engineer the interim analysis results from the updated sample size. This would be a very important component of protecting trial integrity in an adaptive design setting. And for pretty much the same reasons, the data-driven decision rules that would be applied in interim looks must be prospectively specified. All aspects of the statistical rules for increasing the total sample size, for example, must be set in stone. If the trial sponsor or members of the independent monitoring committee are allowed to override the predefined rules and introduce new rules on the fly, that will definitely compromise the credibility of the trial's results. And in fact, there have been some examples of phase 3 confirmatory trials where the sponsor was not careful enough about maintaining trial integrity and enforcing those predefined decision rules, and the United States Food and Drug Administration ultimately refused to accept the trial results. The regulatory agency essentially communicated to the sponsor that they would have to conduct another confirmatory trial before regulatory approval could be granted because they violated some of the key requirements for conducting adaptive trials. Let's talk briefly about clinical trial simulation in the context of adaptive designs. We've already touched upon this very important topic. And to understand what adaptive design brings to the table in very tangible quantitative terms, for example, to support a meaningful comparison with traditional designs that have no data driven decisions, we have literally no other means but to turn to clinical trial simulation. With a simulation based approach, we can quantify potential advantages of not just one but perhaps several candidate adaptive designs in each particular setting. And by doing this, we will model the patient enrollment process, the patient dropout process. We can estimate the timing of the individual interim looks. We can compute probabilities of important outcomes. For example, what is the probability of modifying the trial design? And of course, the list goes on. All of those key characteristics of an adaptive design could be assessed quantitatively only by utilizing clinical trial simulation, and regulatory agencies fully recognize the importance of clinical trial simulation. When it comes to adaptive trials, as you can see on the slide, there's even a section on simulations in adaptive trial planning in the FDA guidance on adaptive designs. And what are the general objectives of clinical trial simulation in the context of adaptive trials? First and foremost, it would be most helpful to estimate the most important characteristics of any trial. In the context of phase 2 trials, that would be to evaluate the type 1 error rate, the likelihood of an incorrect conclusion, as well as the probability of success or power and other important operational characteristics. And when we look at the general goals for clinical trial simulation for confirmatory phase 3 trials, those simulation exercises typically focus only on estimating the probability of success or power, as well as other operational characteristics that I've mentioned. For example, the likelihood of modifying the trial design. Explicitly, the alpha control is not established because the requirement for confirmatory phase 3 clinical trials is that the type 1 error rate control must be established using analytical methods. It will be very important for us to introduce key statistical methods used in adaptive confirmatory trials or adaptive phase 3 trials. In this section, I'm going to introduce concepts that play a central role across different types of adaptive designs, including the 3 types of adaptive designs I defined earlier in this module. So once again, I'm going to introduce the concepts and provide quick examples. Of course, technical details will be beyond the scope of this training program. Our goal, as we said before, will be to avoid most technical details while at the same time aiming at a conceptual understanding. I will share the references that you could review if you would like to learn more about the statistical concepts and methods used in adaptive designs. The references are available on the training program's website. And there are 2 different types of statistical methods I'm going to introduce in this section. The first 3 pertain to the process of designing adaptive trials. That includes conditional power, combination function principle, and a closely related, actually mathematical equivalent to it, conditional error function principle. In addition, in certain types of adaptive designs, such as adaptive designs that support data-driven treatment and population selection, we will have to account for multiplicity induced by multiple hypotheses to be tested in a trial, and to construct reliable multiplicity corrections or adjustments, we will need to rely on the so-called closure principle. We will begin with conditional power, And to set a general foundation for adaptive designs, it is very helpful to begin with conditional power. It is one of the most fundamental concepts, most commonly used concepts in adaptive trials. And in very simple terms, it quantifies the predicted probability of success or projected likelihood of success given the results, the findings available at the time of an interim assessment. Conditional power is derived using a frequentist statistical framework, but it is not that difficult to construct conceptually similar quantities or predictions using a Bayesian framework. And in that case, it is typically, those quantities would be typically called conditional probability. And what is most important here is not really the statistical framework, but the ultimate objective of predicting the final outcome in a trial to inform go no go decisions or other adaptive decisions at the time of the interim analysis. And when I say go no go decisions, it is the terminology commonly used in pharmaceutical drug development, and it typically refers to futility assessments. For example, if there's a little evidence that the treatment or intervention has a beneficial effect, it would no longer be worthwhile to continue the trial. And how do we define little evidence in the context of this set of go no go decision rules? Very commonly, this assessment is made using the predicted probability of success in a trial or a conditional power. If the predicted probability of success is low, for example, it is less than a 10% threshold or a 20% threshold, then it would be futile to continue the trial. This prediction can also be used for other purposes. For example, it could be used to justify a decision to increase the total sample size, or the target number of events in an adaptive trial. Again, we'll go over those examples in detail later in this training program, but suppose we have a clinical trial or a general study that was initially powered at 80%, and the review of interim analysis data reveals that the predicted probability of success based on conditional power is only 70% based on the initial sample size. In this case, what the trial sponsor could do is they could consider increasing the sample size to achieve the desirable level of that predicted probability of success, to increase it, for example, up to 80%. That is, in fact, the key idea behind sample size re-estimation rules based on conditional power. And if you would like to find out more about the use of conditional power, the underlying statistical properties of this quantity, I would like to recommend the section in a great book by Professors Wasmer and Brandt that was published in 2016. You see the reference at the bottom of this slide. On this slide, we will talk about the combination function principle, which is the main statistical tool used in adaptive designs. To understand why this principle plays such an important role in adaptive designs, for example, adaptive designs with sample size re-estimation, event count re-estimation, we need to keep in mind that an important feature of data-dependent changes based on unblinded interim data, for example, an updated sample size, is that data-dependent changes would contribute to alpha inflation. The type 1 error rate would be inflated, and as we discussed that a few minutes ago in this module, general scientific principles, regulatory principles require that appropriate statistical methods should be applied to guarantee alpha control in confirmatory clinical trials. And that's exactly where the combination function principle comes in. It is the most popular statistical method that guarantees alpha control in adaptive trials with sample size re-estimation. And I will provide more information. We'll walk you through the process of applying this principle to multi-stage adaptive trials that enable sample size re-estimation. And once again, if you'd like to find out more about this principle, you could review section 6.2 in the same book on adaptive designs in clinical trials. Here, I would like to very quickly mention that just for your general information, there's another principle that could be applied in adaptive trials with a sample size re-estimation. It is known as the conditional error function principle. It was introduced independently of the combination function principle by two American statisticians in 1995. And you may come across adaptive designs that are set up using this principle, but it's important for us to remember that from a mathematical perspective, this principle is actually equivalent to the combination function principle, and this is all we need to know about it. We're not going to come back to this principle in this training program. The last principle for us to discuss is the closure principle. It is another fundamental principle used for setting up methods for multiplicity correction or multiplicity adjustment. Sometimes we refer to them as multiple testing procedures. All modern multiple testing procedures have been built, have been defined using this principle. And when do we apply the closure principle in adaptive trials? As I explained a minute ago, the combination function principle is applied to address potential alpha inflation that could arise in clinical trials, in adaptive trials with data-driven decision rules such as sample size re-estimation. In this case, there are no other sources of multiplicity, and therefore there is no need for the closure principle. And in part 1, when we get to talk about adaptive trials that only enable sample size re-estimation, I will not mention the closure principle. But later on in this training program, in part 7 and 9, specifically, more complex trial designs will be considered. I'm going to present classes of adaptive designs with the data-driven treatment selection, part 7, and then population selection in part 9. And in the context of those adaptive designs, there will be multiple treatments, multiple interventions, or perhaps multiple patient populations that will need to be assessed, and this factor induces multiplicity even if a traditional setting with a fixed sample size is assumed in the trial, because there will be multiple shots on goal. We will get to decide, for example, whether or not only the best treatment arm should be retained for the second stage in the trial. And in this case, the combination function principle would not be sufficient for this class of adaptive designs. In this case, to address alpha inflation, we will need to invoke the closure principle. And the same logic would apply to adaptive designs with population selection that we will talk in part 9. In a multi-population setting, a statistically significant effect could be potentially claimed in any of the multiple patient populations, in the overall population of patients, or in any of the predefined subsets of that population, in which means that we have to deal with multiple shots on goal. And in this case, we will have to rely on the closure principle to ensure alpha control. To find out more about the use of the closure principle and multiplicity adjustments based on this principle, I would like to recommend the chapter from a book that I published with my colleagues in 2009. We are done with the review of fundamental statistical principles and methods that are used in adaptive designs, and we're now ready to move on and consider the most important classes of adaptive designs employed in a confirmatory setting. This would be a class of designs that utilize unblinded interim data to update the sample size or target number of patients or target number of events in a clinical trial. And these designs, as we have said before, they're known as sample size re-estimation designs or event count re-estimation designs. And this last this last class of designs, of course, would be relevant if the primary endpoint is a survival or similar endpoint. This approach to designing adaptive trials will be illustrated using the VALOR trial. It is a great example of a successful application of general principles of data-driven adaptation. It would be helpful to begin with some background information. The VALOR trial was conducted a few years ago, about 10 years ago. It was a randomized, controlled clinical trial in patients with relapsed or refractory AML or acute myeloid leukemia, and it was designed to investigate the efficacy and safety of an experimental treatment plus chemotherapy versus chemotherapy alone. This was an oncology clinical trial, and therefore it was designed as an event-driven trial. The primary efficacy endpoint here was overall survival, which means that was defined formally as the time from randomization to death of any cause, or potentially censoring if patient happens to be alive at the end of the trial. What is most important for us here is that this trial incorporated an adaptive design, and a justification for utilizing an adaptive approach is shown at the bottom of the slide. In very simple terms, the experimental treatment was expected to provide improvement over the selected chemotherapy, but it was not clear up front how strong the beneficial effect was going to be. The sponsor did not have a good estimate of the hazard ratio that should be used in the process of computing the target number of events in the trial if this was to be a traditional design. And this is something that would apply, of course, to most adaptive designs that are aimed at sample size re-estimation or event count re-estimation. In this case, we will talk, of course, about re-estimating the target number of events because it's an event-driven trial. And the probability of success here depends on the number of observed deaths as opposed to the number of enrolled patients. So if the sponsor is interested in improving the probability of success, given the interim analysis results, they would increase the target number of events in the trial. And this kind of approach would help the trial sponsor to efficiently address uncertainty around the expected effect size, in this case, the expected hazard ratio in the trial. Here is some general information on the decision rules that describe the planned design modifications in the VALOR trial. And as we have said before, it's very important for us to emphasize that all of the design modifications must be pre-specified, must be pre-planned, because here we're dealing with a confirmatory setting. With the original design, traditional design, about 375 events would be required to achieve 90% power in the trial. And within the adaptive setting, a two-stage design was employed, and that involved a single interim analysis. This was the milestone that separated the two trial stages. That interim analysis would be taken after 173 events were accrued in the trial, which is, as you can see, roughly 50% of the target number of events. And the following decision rules were to be employed at that interim analysis: the futility stopping rule as well as the efficacy stopping rule. Those will not be discussed here. We're not going to emphasize them here. We're going to emphasize the rule for updating the target number of events. Based on the unblinded interim data in the trial, the sponsor, using conditional power, using the predicted probability of success in the trial, could potentially increase the event count by up to 50%, which means that up to the total number of 562 events, in an attempt to improve the probability of success if conditional power turned out to be less than 90% at the time of the interim analysis. And Here, maybe before we move on to the next slide, I will make a general comment that instead of the term multi-stage design, for example, a two-stage design, you may also sometimes come across the term stage gating approach in clinical trial publications that deal with adaptive trials. And I think it's a good descriptive term because each stage in this case does serve as a gate, as a kind of a gatekeeper for the next stage of the trial. And we need to pass that gatekeeper to be able to proceed to the next stage of the trial. This gatekeeper could represent a futility assessment, as it was the case in this trial. And then at the end of the first stage of the trial, the sponsor may discover that, for example, the efficacy signal is not as strong as initially expected, and then in that case, the best course of action will be to discontinue patient enrollment, terminate the trial. So, which means that in this case, the sponsor would be unable to pass the gatekeeper. This slide, as well as the next slide, provide a visual summary of the decision rules employed in the VALOR trial based on the predefined two-stage adaptive design. Specifically, this particular slide shows the data available to the sponsor, or technically to the external independent data monitoring committee at the end of the first stage. We can see here that the data monitoring committee reviewed the data from the control patients as well as the patients allocated to receive the experimental treatment at the time of the interim analysis that was performed after the 173rd event, or the death, because the primary efficacy endpoint was overall survival. And the data, the interim analysis data, were to be used to support a number of decisions, but here we're going to focus on a decision to potentially increase the number of events in the trial if the efficacy signal or conditional power happens to be lower than originally expected. Two options were considered at the time of the interim analysis. The first option, in case the efficacy signal was sufficiently strong, would be simply to continue with the original design, which was to accrue an additional 172 events, so that the total would be 375 events. And that would guarantee that the overall probability of success in the trial would be at least 90%. Another option, and this is what actually happened in the VALOR trial because the efficacy signal turned out to be weaker than anticipated, would be accrue up to 379 additional events to the total of 572 events if conditional power was indeed lower than 90%. In the VALOR trial, conditional power level turned out to be sufficiently low, and the sponsor decided to go all the way to the predefined cap. The total number of events was indeed increased up to 572 events. And I would also like to mention an important operational detail that would apply to this particular trial, as well as a general adaptive trials that support event count re-estimation. Theoretically, when a decision is made to increase the number of events in a time to event design, there is no need to enroll more patients because this will not affect the probability of success in the trial. It is only driven by the number of events. But operationally, it would generally make sense to enroll additional patients because this will help keep the trial reasonably short. And in the VALOR trial, when a decision was made to increase the target number of events all the way up to 562 events, in addition, a decision was made to enroll 200 additional patients. And at the end of the trial, I think that's a very important point here that we'll need to discuss, is that the sponsor emphasized that what they really appreciated about this adaptive design. It was the fact that this adaptive approach helped them mitigate the risk of initial overinvestment, What this means is that at the design stage of the trial, the sponsor could have potentially adopted a more conservative approach. They could have considered a larger trial with the goal of accruing 562 events. But this could have been potentially an overkill. The treatment effect, the overall survival benefit, the hazard ratio could have been actually much stronger than initially projected, and as a result, a lot more patients than necessary would have been enrolled into the trial. This essentially creates an ethical problem of exposing much more patients than necessary to chemotherapy, which was the control that is known to be less than ideal in this patient population. And instead of this, the trial sponsor chose a more pragmatic and ultimately more scientifically appealing, more scientifically sound approach of designing a smaller trial, but including an option to examine unblinded interim data to potentially modify the design. But that modification would be considered only if the interim data suggests that it would be helpful to recruit more patients and re-estimate the target number of events. So it's one of the reasons why adaptive designs in general, and specifically adaptive designs that support sample size or event count re-estimation, have become so popular in pharmaceutical drug development as well as in general studies. We're now ready to continue to the next class of adaptive designs. This would be adaptive designs that enable efficient data-driven treatment selection at an interim analysis. And when we discuss adaptive treatment selection designs, it is helpful to mention the so-called platform trials. I know it has become a very hot topic, especially in the world of oncology, and there's a lot of interest in potentially designing platform trials that, in simple terms, simply represent multi-arm and multi-stage trials. And what this means is that these are adaptive trial designs with multiple trial arms or interventions, which corresponds to the multi-arm part of the definition. And those interventions, those treatments, potentially undergo multiple interim assessments. That's where multi-stage comes from. And more complex platform trials are most commonly used in proof of concept or phase 2 trials. When we focus on phase 3 trials, much more straightforward multi-arm, multi-stage designs are considered, and they essentially simplify to adaptive treatment selection designs with one or at most two interim assessments that are aimed at futility stopping rules, at futility assessments, as well as identifying the most promising dosing regimen out of the initial set of dosing regimens. This is based on predefined treatment selection rules, also known as formally as graduation rules. A treatment selection rule helps the trial sponsor decide whether or not they should drop a trial arm, which means halting patient enrollment into this arm, and the other trial arms would graduate, quote unquote, by moving forward to the next stage of the trial. To motivate this approach to designing adaptive trials, I will provide a bit of information on the enhanced trial that we'll use as a way to motivate this approach to adaptive trial design. This was, as I pointed out earlier in this module, one of the first, if not the first, successful application of an adaptive approach to dose selection in a confirmatory trial. This was a phase 3 trial in patients with chronic obstructive pulmonary disease, COPD, and it was set up as a seamless phase 2, phase 3 trial in the sense that it was a two-stage trial. The first stage corresponded to a dose-finding phase 2 trial, and it was actually a 7-arm trial. There were 4 doses of indacaterol, which was the experimental treatment, and they were tested versus placebo as well as 2 active controls. A single interim analysis was employed, and it was used to assess the efficacy of those 4 doses of the experimental treatment to ultimately select the most promising doses that would graduate, quote unquote, to the second stage of the trial, which was the confirmatory stage of this trial. This was, of course, a pre-planned interim analysis, and the goal was to select two best indicatorial doses based on predefined criteria. A visual summary of the key considerations in this two-stage adaptive design are presented on this slide and the next slide. This slide shows us the phase 2 or dose finding, if you will, portion of the study, the stage 1, that includes 4 different doses of the experimental treatment indicatorol that would be compared to placebo, as well as 2 active controls that here are labeled A and B. The interim analysis was performed after 770 patients completed the treatment period, and the interim analysis data, just like in any other ultimately confirmatory adaptive trials, were examined by an external data monitoring committee. And the goal, once again, was to identify the 2 best, most promising doses for the final assessment. This slide shows us what happened in the confirmatory or phase 3 portion of the study using the 2 most promising doses of indicatorol that are labeled here doses 3 and 4. The final analysis compared those 2 selected doses to the passive placebo control as well as the active control. And the most important feature of this type of a 2-stage adaptive design was that all available data, stage 1 and stage 2 data, were pooled for the selected doses of the experimental treatment, and that all available data were used at the final analysis to perform a reliable assessment of the efficacy as well as safety profile of the selected doses to the available controls. And I guess to summarize, this was a huge success story. The trial's outcome was positive. The adaptive design did help the trial sponsor efficiently identify 2 most effective doses of the experimental treatment, and that treatment indicatorol is now approved globally for the treatment of COPD. The last class of adaptive designs that we're going to review in this training program is known as the class of adaptive population selection designs, or sometimes they may be referred to as adaptive population enrichment designs. This class of adaptive designs was actually selected in the FDA guidance on adaptive designs to illustrate attractive features of a general adaptive approach, and I will say a few words about this. It is presented under the rubric Improved Understanding of Drug Effects. In this section of the guidance document, the guidance argues that certain types of adaptation enable clinical trial sponsors to address questions that they cannot answer with traditional or non-adaptive designs. An example would be to consider an adaptive population design with 2 predefined patient populations, which would be the overall population of patients that have the condition of interest, as well as a subset or subpopulation, sometimes known as the targeted group. For example, a subgroup of patients with a positive biomarker status, and those patients are expected to experience enhanced efficacy compared to patients in the complementary subgroup or patients with a negative biomarker status. And in this case, in this multi-population setting, in a traditional design, the sponsor faces a dilemma of selecting the right patient population for the trial. For example, should the sponsor enroll all patients with this condition of interest, or should they focus only on the predefined subset of patients? Or should the sponsor conduct 2 different trials, one for each population, which most likely will be prohibitively expensive? And with an adaptive approach, the trial sponsor could actually fold those two objectives into a single trial. They could incorporate one or two interim assessments to examine the magnitude of the treatment effect within each population and then select the best, the most promising subpopulation for the final assessment. All of this could be accomplished in a single trial, and this is exactly what adaptive population selection designs are about. That's exactly the framework I'm going to present in this section, and we're going to come back to this in part 9. And let me say a couple of words about the case study we're going to use in this section. It is based loosely on the famous Saturn trial. This was the first trial that employed a multi-population paradigm, which means that two patient populations were prospectively defined in the trial, and they were evaluated at the end of the trial. But an important point for me to make now is that this trial relied on a traditional design without any adaptive data-driven decision rules. And the reason I'm going to use this trial here is because there are currently really no good publications on adaptive population selection designs. So going back to this case study, what were the objectives of the Saturn trial? This was, as you can see, a phase 3 trial for the treatment of advanced non-small cell lung cancer, and there was an investigational treatment, erlotinib, that was compared to the standard of care, to the control, and the primary efficacy analysis was formulated in terms of progression-free survival, and the experimental treatment was expected to prolong progression-free survival, which means prolong the time from patient randomization to disease progression. A very important feature of this trial was that a potentially predictive biomarker was specified at the design stage. This was a biomarker based on the so-called epidermal growth factor receptor, known commonly as EGFR, and patients were classified at the time of randomization as EGFR positive, or we can simply refer to them as biomarker positive, and that was the case if their tumors had overexpression of the EGFR protein, and if this was not the case, then they were classified as EGFR negative or simply biomarker negative. And it was known that the biomarker positive status could potentially be predictive of treatment response through directly through the mechanism of action, because erlotinib was an EGFR inhibitor. So patients with a positive biomarker status were expected to experience enhanced efficacy, longer time to progression, disease progression, compared to patients in the complementary biomarker negative subset. And to account for this multi-population design, the treatment effect, the hazard ratio for progression-free survival, was evaluated at the final analysis not just in the overall population of patients, which would have been done in a traditional single population design, but it was the final analysis was designed to examine the effect in the overall population of patients, as well as the predefined subset of patients with a biomarker positive or EGFR positive status. Looking at the general justification for an adaptive design, in this particular case, I've already briefly talked about this. As we said, a traditional design without any data-driven elements, any data-driven decision rules was utilized in the trial, but I believe, and that I'm going to illustrate this fact, is that an adaptive design, I believe, would have helped the sponsor of the Saturn trial to pursue two objectives in a single trial much more efficiently. For example, they could have used an interim analysis to estimate the hazard ratio, the magnitude of the beneficial effect within each patient population, and using this information, the most promising population could have been identified for the final assessment. which means that two patient populations could have been easily studied within a single trial. And on the next several slides, we will discuss how an adaptive design with a population selection rule could have been applied in the context of this particular trial. Let's take a look at the following diagrams that are conceptually, of course, quite similar to what we discussed in the context of sample size re-estimation and treatment selection. Here we're looking at the data available to an external data monitoring committee at the interim analysis in this two-stage adaptive design. That first stage data includes control and treatment patients up to a predefined number of primary events, which is the number of patients who experienced a certain number of patients who experienced disease progression in this case. And then an adaptive population selection rule was pre-planned to be applied at that time. And the effects within the biomarker positive and biomarker negative subpopulations were to be evaluated to identify the best patient population for the final analysis. This leads us to two different options. Option one is presented on this slide, which is it is possible that the interim analysis data could have supported the decision to simply continue enrolling all patients after the interim analysis. When would that decision be made? It would be most natural to make that decision if there's evidence of a strong treatment effect within each subpopulation. So this new experimental treatment, erlotinib, may be equally effective for biomarker positive as well as biomarker negative patients. In this case, the final analysis would be performed in the overall population of patients. An alternative approach would be to consider option 2, which would be to enroll only biomarker positive or EGFR positive patients after the interim analysis. And that would be the case. That decision would be easy to justify if it was clear at the interim analysis that the treatment benefit is clearly limited to the subset of EGFR positive patients. In this case, as you can see here in the diagram under stage 2, only biomarker positive patients would be enrolled after the interim analysis, and naturally, the final assessment will only be performed in the subpopulation of biomarker positive patients, which leads us to the following conclusion or summary that the Saturn trial had a successful outcome based on a traditional design approach, but it could have benefited from a design that employed adaptive population selection. And there are multiple other trials I have worked on that took advantage of interim decision rules for population selection. And we're going to come back and dive much deeper into this topic later in this training program. As I pointed out before, in parts 5, 7, and 9, respectively, we will talk about the relevant examples, case studies, and the underlying statistical methodology and operational rules for adaptive designs that support 1. Sample size re-estimation, 2. Treatment selection, and finally population selection. We're almost done with part 2 and we are ready for conclusions. The conclusions are presented on this slide as well as the next slide. We will begin with a general discussion of the reasons why adaptive designs have become so popular in confirmatory or phase 3 clinical trials, because they do help trial sponsors efficiently address inherent uncertainty at the design stage. For example, there could be substantial uncertainty about the anticipated size of the treatment effect, as in the VALOR trial, and the sponsor could then address that, mitigate that risk of improperly sizing the study by employing an adaptive design with an option to increase the number of events or the sample size if the interim data suggest that this increases adjustment would be beneficial. For this reason, the general adaptive design approach have become very popular in confirmatory trials, and adaptive designs of the kind that we have discussed in this module have been successfully applied to hundreds of phase 3 trials. I would like to point out that, and we'll come back to this point again and again, is that there are certain restrictions, that certain conditions that would make an adaptive approach more viable in a particular setting. And one of the key assumptions is an assumption of a slow patient enrollment. In general, benefits of an adaptive approach are maximized when patient enrollment is fairly slow. And I would like to say also a couple words about the general regulatory requirements or general scientific guidelines that are applicable to a very broad class of adaptive designs. We went over them in this module, and it's very important for a sponsor to address issues related to operational bias, to take steps to maintain trial integrity. For this reason, for example, an external data monitoring committee would be required. And given all of those additional complexities involved in the process of designing of adaptive trials compared to a traditional setting, it is a recommendation, it is almost a requirement to rely on clinical trial simulation to evaluate key characteristics of an adaptive design that will help, first of all, justify a decision why an adaptive approach should be considered in the context of a particular development program or a more general study, and secondly, to quantify key characteristics, estimate key characteristics of the adaptive design. And throughout the rest of this training program, we will definitely set aside time to go over the available software tools that facilitate the process of clinical trial simulation and help answer those very important questions. I appreciate your interest in this online training program. I would like to encourage you to watch the other videos and share information on this training course with your colleagues. Thank you very much.